Okay, in this video we're going to take a look at instincts and genes. This is part 2 of our lecture 8. Let's move to the PowerPoint slide. Oops. We've covered these. So genetics and instincts, if you will. And I wanted to go back to talk about the mice, the mice nurturing behavior. It turns out that mice have very specific nurturing behaviors. When a female is ready, mature, she makes a nest of straw or feather or fur in a safe place. She gives birth to a number of different young, right? The pups, they're called. And she bites off their amniotic sac, she eats the placenta, and then she gently picks the pups up and carries them to the nest, where she begins to nurse them, and she keeps them huddled. Okay? This nurturing behavior in female mice is considered instinctive. It's direct, it's stereotyped, it's reliable behavior, and it unfolds gradually as the mice become mothers, if you will. There doesn't seem to be a learned component to it. It seems to be instinctive. Okay? Well, there are some mice that researchers have created that have a mutation in a particular gene called the FOSB gene. And these mice are actually, we use this language they're called knockout mice when they've lost the functioning of a particular gene. So researchers created these knockout FOSB moms, if you will, all right? And they exhibited basically very normal behaviors in many, many ways. So they are cognitively intelligent. They have normal sensory systems and respond to sensory things very normally. But it turns out when they gave birth, all of their pups born to these knockout FOSB moms died within a day or two, either of cold or starvation. Well, what was happening? And by the way, these pups were not sick or anything. If pups from these knockout moms were given to foster mice moms, they did just fine. So it's not that the pups were somehow unhealthy. The moms simply didn't care for the young, right? The young died of cold or starvation in 1 to 2 days after birth. The moms didn't pick them up and carry them to their nest. They didn't huddle them. Otherwise, they seemed very normal. The moms were making milk. The moms were had a normal sense of smell, a normal sense of cognition. Just didn't do the nurturing behavior. What's happening in these knockout FOSB moms? Okay. Well, something that initiates maternal care or nurturing is missing. She isn't responding to the appropriate environmental stimuli. The releasers here, the sign stimuli, if you will, for nurturing behavior should be the pups, the little helpless pups that are just born, right? So it seems that this gene is responsible for a link between brain and behavior, right? There's some sort of signaling that this gene produces. It's expressed when mom smells the pups. And the FOSB protein actually is a regulator of other genes. It turns them on. It's a transcription factor that regulates other genes and sensitizes the hypothalamus. And the hypothalamus is involved in all sorts of regulatory behaviors, including things like nurturing behaviors. So the presence of this gene mutation prevents instinctive behaviors from happening. When this gene is mutated, the instincts don't work. And researchers found that experience didn't help these mice. So they couldn't really learn this after, seeing a bunch of their first pups die, tried again. Didn't matter. Their second batch of pups, litter of pups, also died. Their third litter of pups also died. So it wasn't that they could just gain some knowledge from experience. This is an instinctive behavior that is in huge part controlled by the protein product of FOSB gene. That's just a kind of cool example. We've seen it before. Here's another example that we've kind of touched on before as well, and that's prairie voles. Okay, prairie voles are my favorite. I think I would have studied them if I went back to school today. We've talked about them, but let's review them and think about genes and instinct. So we know that prairie voles are monogamous, at least socially monogamous. They form pair bonds, and they take care of the young males and females together, okay? And their relatives, montane voles, are polygamous. The males will mate and leave, and there's no pair bonding that happens at all, right? In prairie voles, the hormones oxytocin and vasopressin seem to be important in eliciting this pair bonding behavior. Okay, we think this pair bonding behavior is instinctive. It doesn't need to be learned, and it's stereotyped. It's typical in the whole species. It seems to be innate. It's an instinct, if you will, and it seems to be in part controlled by oxytocin and vasopressin. Now, what did Insel and Shapiro do in 1992? They investigated the brains of prairie voles and montane voles, these polygamous montane voles. And here's what they found. They found no difference in the expression of oxytocin and vasopressin in the brains of these voles. So there was the same amount of oxytocin and vasopressin in the same places in these voles, but there was a huge difference. The prairie voles had many more oxytocin receptors in certain parts of the brain than did the montane voles. Okay, so certain parts of the brain had an abundance of oxytocin receptors in prairie voles that weren't there in montane voles. And the presence of those receptors, it seems, leads to the pair bonding, if you will. So what Insel and Shapiro did was they injected oxytocin into prairie vole brains, okay, or into montane vole brains. When they injected the oxytocin into the montane voles, it didn't elicit any kind of monogamous behavior or pair bonding at all. But it did induce monogamous behavior in the prairie voles. That's no surprise. That's what we would expect. But what they found was that if these prairie voles had oxytocin receptors that were blocked, then the prairie voles didn't show any of the pair bonding behavior either. So it's the presence of these receptors that are in certain regions of the brains that allow prairie voles to respond to the oxytocin and then form pair bonds to show this instinctive behavior. Are there any particular genes that we can find that would contribute to differences in the receptor locations, right? Or in the abundance and the distribution of hormone receptors, right? Well, they looked at the montane voles and the prairie voles, and they said, all right, we're going to look for genes for these oxytocin receptors. It turns out they were identical. Identical genes in the prairie voles and the montane voles. But, and here's the kicker, the promoter regions for these genes differed between prairie and montane voles. The prairie voles had, in their promoter region of the genes for oxytocin receptors, it's a lot to say, they had extranucleotides, a whole bunch of extranucleotides in the promoter. Somehow, having a different promoter region of this oxytocin receptor gene contributes to very different instinctive behaviors. That's pretty fascinating, where you can link a single gene change to very different not nurturing behaviors, going back, I was going back, to very different pair bonding behaviors in our prairie voles. Now, we have to study exactly how that works. And we want to look at, why is it that a difference in this promoter region triggers a difference in the distribution of the receptors and then a difference in nurturing behavior. It's pretty cool, though. It's a pretty cool connection, if you will. So those are just a couple of examples of how genetics can be connected to these very innate, instinctive, stereotypical, but often complex behaviors in animals like prairie voles or herring gulls or stickleback. Okay? In the next video, we'll talk briefly about fear behavior in humans and in other primates. Alrighty.